Hello everyone, welcome to my classroom by Ms. Sejal Patel. Today's topic is cleaning validation.
What is cleaning validation? Cleaning validation is the methodology used to assure that a cleaning process removes chemical and microbial residues of the active, inactive, or detergent ingredients of the product manufactured in a piece of equipment, the cleaning aids utilized in the cleaning process, and the microbial attributes. Introduction: cross-contamination must be avoided in the pharmaceutical industry at all costs, and successful cleaning validation ensures that patients are not put at risk due to cross-contamination. The process can be divided into a number of sections, each of which must be fully understood and areas of concern addressed to ensure a successful outcome across the entire process. This spans both the manufacturing and subsequent analytical and microbiological support. The data used to confirm a positive/successful cleaning validation is underpinned by the results of validated analytical methods. It is essential that these results are truly representative, as patient safety is based upon the absence of equipment residues. So what are those areas of concern? What affects your ability to get a successful outcome, and what do you need to consider when carrying out a cleaning validation exercise?
An overview of the areas to be considered to ensure success.
Areas of concern: 1. Service lines. 2. Gaskets. 3. Filter meshes. 4. Valves. 5. Pumps and samplers. 6. Process investigations. 7. Packaging lines. Design and control of cleaning methods. Good cleaning procedures are fundamental. A procedure that has been used for years may not necessarily be the best. A review of the methods should be carried out at validation to determine their suitability and effectiveness. Procedures should include factors such as 1. Operation of valves during cleaning. 2. Filling pipework with solvent and allowing dissolution time. 3. Refluxing solvent around system and through condenser return lines. Issues with sampling and testing. A number of issues can be identified related to the swabbing procedure, and these must all be addressed if success is to be guaranteed. Control of swab equipment. It is essential that all the equipment used to carry out a swab test is reliably controlled. It is extremely easy to contaminate a swab and put your whole cleaning validation exercise at risk of failure. The swab material, the solvent used, any disposable gloves, etc. must be very carefully controlled to reduce the risk of possible false results. Being able to determine the correct area to be swabbed even when they cannot visually see where the task is being performed. Failure to swab the required area will result in either a failed swab or, worse still, from a cross-contamination viewpoint, a past result which should really fail. Their reproducibility in being able to repeat this exercise 4 times in exactly the same place. It has been demonstrated that a swab taken using 4 wipes of the same area results in maximum recovery, greater than 90%, from a given surface. However, the ability to do this requires considerable skill and training. Development and validation of analytical test methods. Determining cleanliness can prove to be a very challenging task, with the aim being to measure trace residues, target analytes, on the surfaces. The residue must initially be extracted from a surface, recovered from the extraction matrix, and then suitably quantified. Note that sampling is usually performed via direct methods, swabbing. However, indirect strategies, such as rinse samples, may also be used, though the latter is not a preferred approach. Very often, the starting point in the development of a cleaning method would be based upon a previously developed assay method. In many respects, the requirements for validating cleaning methods are closely aligned with a standard assay, evaluating factors such as specificity, accuracy, recovery, linearity, sensitivity, precision, robustness, and solution stability. Swabbing. Often, insufficient time is invested in the actual swabbing regime, which can ultimately lead to a less than robust final method. Each of the surface types identified, discussed previously, should be spiked with the API at the calculated residue limit, and experiments performed to demonstrate that acceptable recovery is repeatedly obtained. This can involve the evaluation of a number of different swabbing solvents and swab types together with optimization of the actual swab technique. This, combined with the need to be able to repeatedly detect residues at very low levels, can contribute significantly towards the time required for successful method development and validation. Microbiological contamination. Aside from the manufacturing and analytical aspects, it is also paramount to consider cleaning validation from a microbiology point of view. This focuses on contamination control in pharmaceutical and healthcare facilities. Pharmaceutical manufacturing process can be subjected to microbial contamination. Therefore, a good cleaning and disinfection process is necessary to maintain a good hygiene in facilities. Disinfectants are used to prevent microbial contamination of pharmaceutical products. Where chemical disinfectants are used as part of contamination control, it is important to evaluate the efficacy of the disinfectants to ensure they are fit for purpose. Choosing a disinfectant. Disinfectants are usually chosen based on their chemical properties and expected effectiveness against microorganisms, bactericidal and fungicidal agents, sporicidal agents, sterile disinfectant. Microbiological practical test. The efficacy of a disinfectant can be demonstrated with a suspension test or a surface test. Suspension test can be used to determine if the concentration at which a disinfectant is used is effective. Since the disinfectant is tested in a suspension, it does not mimic the way it is used in practice. A surface test is designed to mimic a possible contamination on a surface which can be found in a pharmaceutical or healthcare facility in the presence of an interfering substance. The quantitative surface test EN 13697 can be used to validate the efficacy of a chemical disinfectant. Validate environmental monitoring media. To have confidence that your selected disinfectants are not influencing the recovery of organisms on media used to perform routine environmental monitoring, it is important to verify the process. This validation will enable pharmaceutical and healthcare facilities to implement the use of adequate contact plates and swabs containing the appropriate neutralizers in the environmental monitoring process. I hope you find the information useful. I would appreciate your likes, share, subscribe.